Welcome, everybody, to this UCL lunch hour lecture on the changing shape of DNA. My name is Dr. Chris Morris, and I'm Vice Dean for Innovation and Enterprise for the Faculty of Life Sciences at UCL. Before we begin today's lecture, we'd like to briefly recognize that 2026 marks UCL's bicentenary. It's an important moment for us as a community, and to open the event, we'll share a short film that reflects on this milestone. After that, we'll move straight into the lecture.
200 years ago, there was no university in London, and we're really marking the coming of a very radical, very different institution to a city that was the largest city in the world.
UCL was to be open to people of all faiths, or indeed no faith.
Women were admitted to the Slade School of Fine Art when it opened at the start of the 1870s, and from 1878 women were able to come to UCL, study alongside men and get a degree, and this was way before any other university was giving degrees to women.
In UCL's bicentennial year, you can expect a major new exhibition, 3 books, installations from renowned artists, and UCL Illuminated, a spectacular immersive sound and light show on our central London campus.
The main core is going to turn into a multi-day theatre with filming, workshop, lights, laughter, comedy, theatre and drama. It's going to be magic.
I'm really excited about telling people the story of how we as an institution have evolved over 200 years and how we're going to evolve for the next 200 years.
It's now my great honour and pleasure to introduce today's speaker, Professor Zoë Waller. Zoë has an MChem in chemistry and pharmaceutical and forensic science from the University of Bradford, and a PhD in chemical biology from the University of Cambridge. She's a fellow of the Royal Society of Chemistry and a senior fellow of the Higher Education Academy. After establishing her career at UEA, Zoë joined UCL in 2020 and is currently a professor of drug discovery and also the head of the research department of pharmaceutical and biological chemistry within the UCL School of Pharmacy. Zoë's received many awards for both her teaching and her research, including at UCL, a Provost Teaching Award, the Royal Society of Chemistry, Chemical Biology and Bioinorganic Group Lectureship, and the Royal Society of Chemistry Jeremy Knowles Award. Zoë's research interests are centered around the structure and function of DNA, and her research is currently supported by the BBSRC and the Leverhulme Trust. Before we begin, I wanted to let you know that we'll have some time at the end of the lecture for questions from those who are watching, and these can be submitted at any point during the talk by going to Slido and by entering the code #DNA. So I'll now hand over to Zoë for her talk. So,
thank you so much, Chris, for the kind introduction, and it's a great honour to be part of this lecture series, especially for the UCL 200 celebrations as well. So I'd like to start talking about DNA, and it's one of my favourite subjects. And I've given a lot of talks to the general public about the work that we do in my research group. And I always start one of these with DNA and what comes to mind. There are many, many different answers to this, and I'm always very excited to hear what people's perspective of DNA is. And over the years, there have been a few common suggestions for what comes up, in addition to, obviously, the scientific ones. And a lot of these centre around DNA testing. So, for example, Angelina Jolie at one point, it was very clear that she had had DNA testing to determine that she has a predisposition to certain types of cancer. So she'd had a double mastectomy and then also had her ovaries removed to prevent her from getting those conditions. And that was a whole era of those suggestions coming forward. There's been a lot of talk about ancestry and about DNA testing around that. And I looked in the news this week to see about what there might be in terms of paternity testing. And apparently Cardi B has some issues about potentially questioning about paternity. So Offset is apparently asking for a DNA test. And thinking back a few years, there was a whole era where television was focused on DNA testing for paternity, and Jeremy Carr was one of those examples. And then finally, I think the other most common suggestion was through crime scene, so detection of DNA at the crime scene and using that for prosecutions. But DNA is an information molecule. It codes for all life on Earth, whether it be your pet cat or perhaps the tomato seeds that you might have already started growing ready for the summer. All life on Earth is encoded in DNA, and it is an information molecule in that aspect. And when we think about humans, our body is made up of many different types of cells, and in each of our cells, we have a nucleus, which is a special compartment where our genetic information is held in the form of chromosomes, which are a combination of protein and DNA. And that DNA is made up of 4 bases or building blocks. So we refer to them as bases, but they're quite often termed the building blocks of life, A, T, C, and G. So these building blocks, these 4 building blocks, are different bases which comprise the code that encodes all different organisms. So the 4 building blocks are guanine, cytosine, adenine, and thymine, and we shorten these to G, C, A, and T. Now, each of these have a special relationship with each other. So guanine likes to pair up with cytosine, and adenine likes to pair up with thymine, and we call these base pairs. And our genetic code is essentially the sequence of these bases or building blocks. And different organisms have different size genetic codes. So E. coli here is very small, and that has a genetic code that's 4.7 million bases long. Yeast is a bit bigger at 12 million. Worms, bigger even so. But it might come as a surprise that humans and mice have very similar sized genomes. It may even come as more of a surprise that newts have a genome that's even bigger than humans, so 18.6 billion compared to 3 billion bases for humans. But out of all organisms on the planet, it's plants that have the largest genomes. And in 2024, this little fern here broke the world record of having the largest genome on the planet with 160 billion bases long. So if you took the DNA from a single cell of this fern and laid it end to end, it would be the same height as Big Ben, which I find amazing that we can cram that much DNA into one single cell. But biology is amazing, and that's one of the reasons why I enjoy my job so much. So I've talked about DNA being an information molecule, and I would like to address what the DNA codes for. And DNA is like a blueprint of our genetic information, and to use it, organisms convert it into a working molecule. So DNA gets converted into a working copy called RNA, and that is then used to then make protein. Now, I quite often like to use an analogy that because DNA is a blueprint and we use a copy to make it, I like to think about it as an analogous situation to imagine you have a recipe book, and I give the example here, Mary Berry's 100 Cakes and Bakes. And if you have that as a signed copy, you want to keep it nice. So if you did want to use it as actually as the recipe book, then if you wanted to make cupcakes, for example, then rather than reading and using that in the kitchen, you might make a photocopy of the recipe for the cupcakes and then use that to then make your cupcakes. And that's essentially what our bodies do. They use the DNA to then make a working copy, RNA, and then that is then used to make the protein. We've talked about DNA relating to hereditary and inheritance, and DNA is inherited, and we can see that in our families, and it's quite easy to work out here that one of these photos is older than the rest, and this is me when I was about 18 months old. You can tell it was probably from the 1980s because nobody would dare put their children in velvet like they used to back then. And alongside that photo of me is a picture of each of my sons at approximately the same age. And you can see very clearly that there are similar features. Two of them, Eddie and Griffin, have a very similar smile to me. So I've got quite a crooked smile, and they've both inherited my smile. Franklin, although he looks a bit miserable here, he's in hospital in this photo, he has a very different smile. But he does have similar eyes to me. And quite often people will say, your son has your eyes. They're very similar color. But what they actually should say is your son seems to have inherited a gene from you that makes a protein in his eyes that appear the same as yours. Doesn't quite have the same ring to it, but that is essentially what is happening. We look similar because the code that is being used to make us is also very similar as well. So I'd like to now touch on what DNA looks like. And the reason I've used this GIF of the spinning DNA is you'll see it quite commonly used in films. I always like to have a screensaver on a computer in the background. And the amount of times that I see a DNA twirling around is actually quite amazing. So if you look out for it, you'll see it in lots of different films or TV programs. And I describe DNA as a twisted ladder structure. And I regard it as one of the iconic scientific images of the 20th century. And what we mean by this is that if we take something that looks like a ladder and then start to twist it, this is what we get, is like a helical structure of DNA. And then relating this to the different building blocks, the rungs of the ladder relates to these building blocks base pairing. So guanine base pairing with cytosine and forming those interactions between them. Those are represented by the rungs of that ladder. So that hopefully gives you a bit of an insight of how the different building blocks are then linked together. And this is the structure that was first proposed by Watson and Crick in 1953. And again, as I've already said, it is an iconic discovery and an iconic structure of the 20th century, an image that a lot of people will come and say double helix when you mention DNA. But it's quite often, there's a bit of misinformation sometimes, that sometimes people will conflate the structure of DNA and the first proposal that DNA is a double helix with the discovery of DNA. And at one point on Google, if you typed in who discovered DNA, the highlight answer at the top of Google would be Francis Crick. But that's not true. It is correct now, and I think I was using it actually in my teaching for a little while of why you needed to double check things when you search for them on the internet, but it is correct now. But so who did discover DNA? So let's start there. So we need to go back a little bit further to the 1870s, and it was Frederick Miescher who is attributed to discovering DNA. And he spent his days extracting pus from bandages from his local infirmary. And he extracted something which he termed nuclein, which we now know is actually nucleic acids or DNA. And if you're in Germany, you can actually go and see some of his samples in the museums there. So Frederick Miescher discovered DNA. Watson and Crick are the ones that proposed a structure that ended up being what was regarded as correct. So this is what we think DNA exists in most of the time, is this double helical twisted ladder structure. But soon after the 1950s, it became apparent that perhaps DNA can do more than that. And in particular, in sequences that contain a lot of guanine, so the base G, it looked like it could form perhaps something different. And when there is a lot of the base guanine in a particular DNA sequence, it can form something that isn't the same as the Watson and Crick base pairing. It could do something slightly different, and it can form these square shapes, and we call this G quadruplex DNA. These form when guanine does something different to its normal base pairing with guanine. So here's guanine here, base pairing with cytosine, and this is what we would call a Watson-Crick base pair. But guanine itself can actually base pair or start to hydrogen bond with itself. And I like to think of guanine in this case a bit like a corner. So it's got one face where it can provide one type of interaction, and then another face here where it can provide a complement. So, if we go and put another guanine here, you can see you can get some nice interactions there. This happens again and again. So guanine can actually self-assemble into these squares. It can actually also form helices. And if you take guanosine, which is the base component, guanine, and dissolve it, even on its own, it will form a gel, which shows that you can have this self-assembly into a higher order structure. And this is very, very interesting, because if you take one of these squares and kind of twist it so we're looking at this end on, they can then stack on top of each other. And this is what then comprises these square-shaped structures, or these quadruplexes. So in the 1960s, it was established that G-rich sequences or guanine-rich sequences can form G quadruplex DNA. What do these sequences look like? So if we bring this up so it's a bit more clear and we can see all of the different bases, this is what a G-rich sequence will look like. So we've got some guanines and then a little space, some more guanines, a little space of any bases, guanines, a little space, and then some more guanines. So that's what we mean by a G-rich sequence. And then these form these quadruplex structures. Later on, in 1993, it was shown that as well as the G quadruplex structures that form in G-rich sequences, where you have C-rich sequences, they can also form a different structure called an I motif. An I motif comes from the term intercalated, which means that rather than having like rungs on a ladder, like in your Watson and Crick double helix, these are like a crisscross. So if you interlock your fingers, this gives that kind of knotted shape. So cytosine-rich DNA can also form a special type of DNA structure. So here's an example of that C-rich sequence here. Again, we're looking for some cytosines, any bases here, cytosines, any bases, cytosines, any bases, and then some more cytosines. So we need to have 4 sets of these cytosines to form this structure. And then they form this little kind of very, very tightly knotted structure. But hopefully you can kind of appreciate that both of these structures are very different when you look at their shape to Watson and Crick double helix. And one of the things that I wanted to highlight here is that, so in 2002, there was the first disclosure of a crystal structure. So that's we're actually visualizing these G quadruplex structures. So you can see the square shape here. And so this is looking down the G quadruplex. So this was the first evidence of this us being able to visualize the structure. And this work was actually done by UCL colleagues, so Emeritus Professor Stephen Needle, and one of my colleagues that I work alongside, Dr. Gary Parkinson. They were the first to show and prove that these G quadruplexes actually existed using X-ray crystallography. And this came at a time where the Human Genome Project was coming towards the end. And in 2003, there was indication that not all of the DNA codes for protein. So out of 3 billion bases in the human genome, only 2% of that actually coded for protein. And there was a big question as, well, what does the rest do and why is it there? And in 2005, it was shown that 43% of all genes in the human genome contained a DNA sequence that could form a G quadruplex, so one of these special higher order structures that forms in these G-rich sequences. In 2013, these were visualized in cells. So using antibodies, they were able to show that these G quadruplexes exist in cells. It was very highly talked about in the news. It was on the BBC website, etc. So hopefully I can kind of persuade you here that DNA is much more than a double helix. We have our double helical shape, but it actually can form different shapes, G quadruplexes and G-rich sequences, and the I motifs in C-rich sequences. So I guess one of the other questions then is, what do these structures do? And in terms of the Human Genome Project and finding that only 2% of DNA codes for protein, what was realized was that actually quite a lot of DNA was what we call regulatory. So it is important to determine whether genes are switched on or off. So what do these structures do? We think that they provide a place for proteins to interact, to either tell a gene to be switched on or switched off. So if we can encourage this special DNA shape, then we might have a different effect. And that could be useful in terms of development of medicines. So what do we mean by that in terms of reading the code? So if we use our DNA and then convert that to our working copy of the RNA, and then convert that to the the protein, a way of making analogy of this is that if we wanted to run a cake sale, and so we want a lot of protein in this case, one way would be to take your blueprint, take your DNA, make a load of RNA, or in this case, make a load of copies, and then you're able to make a lot of your product. So this in whether it be cupcakes or protein. Essentially, if you're switching on a gene, you're making more of the working copy to then make more of your protein. So DNA is more than a double helix. We have it forming different structures. And before I touch on the work that we do in my research group and the teams that I work with, I want to highlight some other things that DNA can be used for. DNA is and can be used for storage of data, and Microsoft actually used DNA to encode some of their data. DNA can be used for computing, so you can change the shape of DNA, and we've shown that it can form different shapes, and these can be used to do different procedures, and you can have different outputs based on different triggers. So there's a whole area of DNA computing based on that. DNA can be used as a material, so you can form things like, for example, hydrogels out of DNA, and then they will then be able to hold water in them. So it can be used as a material. And there's a whole area looking at making small devices out of DNA, and that's called DNA nanotechnology. So you can make small boxes or cubes or different shapes or different lots of different images. And I think I quite like this GIF because it shows the potential of these different things. Sometimes people might refer to these as DNA robots. And one of the really exciting things about DNA is because you can predict or learn about how you'll have base pairing, you can then design things based on that. So DNA more than a double helix in many, many different ways. So part of the research that I do at UCL is focused on both these structures. I'm kind of known as an expert in eye motifs, and I focus a lot on C-rich DNA. But I feel these structures both interrelate with each other a lot. So the G quadruplex and the eye motif are really important in the work that we do in the School of Pharmacy at UCL. So I talked about DNA a lot, and talked about it being part of cells and being part of the hereditary material. But a lot of the DNA that we work with is actually made synthetically. So we order our DNA through custom oligonucleotide synthesis, and we essentially decide what sequence we want. So here, this is the sequence that we could order here. We ask what synthesis scale we want, how pure we want it, so how it's going to be purified, and any modifications. So we can add fluorophores, like FAM is a type of fluorophore, TAMRA is another type of fluorophore. So this is what we would use in terms of a fret pair, so using fluorescence to identify how folded or unfolded something is. But in the majority of cases, we order synthetically made DNA. And it comes in a little tube, usually something that looks like this. If it was FAM and TAMRA labeled, it would be pink, and you'd be able to see it at the bottom there. But essentially, all of the work that we do on DNA is using custom DNA that has been synthesized in a laboratory, and then we get it in a tube, and then we dissolve it up in water before we do our experiments. And it's usually, I don't know, we spend about £100 for a little smear at the bottom of a tube. But it's exactly what we want, and it's also very highly pure, so we know what we're doing with it. And some of the research that we're looking at is how to change the shape of DNA. So I've showed you that there's this double helical structure, and that you can get G quadruplexes and I motifs. And some of these structures have different triggers. I motifs, for example, they are pH sensitive. And so on a pH range, and you might be familiar with pH in terms of some things are acid, some things are alkali, and I motifs work in a window between approximately pH 5 and pH 8. And so I think these are interesting because this is around physiological conditions. So you can change the shape of DNA using pH. And we've also researched different ways of using metals to change the shape of DNA. And one of the first publications that I had as an independent researcher was looking at silver and silver cations. And we showed that you could take a C-rich sequence of DNA and use silver cations to fold it up into an I motif structure. So this was one of the first examples of showing how you could change the shape of C-rich DNA using metal cations. And then later on, we also were able to do pretty much the same thing using copper-1 cations. And the beauty with some of these metal cations is that they are sensitive to oxygen. So it is actually also possible to convert from an eye motif to another different shape after exposure to oxygen as well. We work a lot on trying to find, in addition to triggers, so we've looked at pH, we've looked at metals, we can try and trigger the change in shape of DNA using small molecules or peptides. And here are some examples of some of the compounds that we've shown that target that DNA. So these are what we would regard as small molecules. And something similar, they may be a bit more drug-like, could be potentially developed into medicines for different genetic diseases. These ones here are slightly bigger. We call these peptides. And you might have heard the term peptide recently because there's a lot of media attention because GLP-1 agonists are based on peptides. So things like Ozempic is they're a lot bigger peptides, so a lot larger than this. But that's what peptides are. These are different types of molecules. And we've shown that these particular compounds and peptides can target eye motif DNA and have biological effects. And we're hoping to develop those into potential medicines to treat conditions and diseases. A lot of the work that I do is in collaboration with others, and actually I'm very lucky to have a lot of people that are very interested in exploring this further. And this is an example of finding molecules that can light up when they bind DNA. So, for example, here is where we have no DNA. There's no light up process, and we add the DNA in, and then we can see a very, very big change here. And this enables us to potentially use these compounds to identify whether we have a particular structure there. So we might be able to use these molecules to visualize or be able to see where we have eye motifs, for example. We're very interested in understanding the shape of DNA. So I've talked about the changing shape of DNA, but really also looking at the detail or the dynamics. And I'm very, very privileged to be able to work alongside a lot of collaborators and then also colleagues that work in the School of Pharmacy as well. So I've given examples here. I work alongside Dr. Gary Parkinson and Professor Josep Hader, and also Dr. Chris Wardby, who are also my colleagues in the School of Pharmacy. And this is an example of some of the work that we published just recently that showed for the first time the structure of an eye motif from the promoter region of insulin. And we're also interested in the dynamics of these structures, how they change. And I also work with Dr. Tim Craggs from Sheffield on that, looking at that using a method called single molecule FRET. We're very interested in how genes can be switched. And actually, and I'm really pleased that Chris is able to be chair for this session because he and I have been collaborators for a very long time. And we're very interested at the moment about how we can change gene expression using peptides or small molecules. And in particular, we've done a lot of work looking at a particular region in the insulin gene and showing that actually, depending on the shape of DNA, you'll get a different response. So here, the insulin gene is only switched on when you have the G quadruplexes and the eye motifs present. And it doesn't if it's forming a different structure. And so we're very, very interested in understanding how genes can be switched on and off using these different structures. And one of the things that we're really interested in looking at is understanding how molecules work and how they interact with DNA. And Professor Stephen Needle, who's a professor emeritus at UCL School of Pharmacy, has a drug that's in clinical trials. It's called QN302. And one of the things that we published recently was looking at how it interacts with different types of DNA. So it targets G quadruplex DNA, but we also found that it also has a different effect on eye motif. So sometimes molecules have a dual function, and I'm really interested in understanding about the dynamics of these different structures. And it's not just about humans, and it's not just about the human genome. I have a lot of interest in how these different structures and how these different DNA shapes can be applied in other organisms. And I have a long-term collaboration with Dr. Andy Gates from UEA, looking at the DNA structures in bacteria, and in particular in those that are in bacteria in the soil that control nitrous oxide emission. So we're really interested in seeing whether we can stop that using different methods as well. So if we can prevent soil bacteria from emitting these potential greenhouse gases, then that would be a really, really different way of approaching that problem. So we're really interested not just in human genomes, but also bacterial and also plants as well. And one of those examples, so Professor Liang Ding is a collaborator where we also look at eye motifs in plants. And one of the things that we have done is looking at how we can try and predict which sequences will form these special DNA shapes. And we recently published an example of this called Eye Motif Seeker. And if you're interested, you can follow and have a little look and try and see which types of sequences might form eye motifs. And this is driven by a machine learning algorithm as well. So you can even feed it more data to enable a better prediction. So what does the future hold? I would really like to look at the more applied sense of what we're working on. So looking at how the shape of DNA affects the progression of disease. We're looking at how it changes during the aging process. And I really am looking forward to developing some of the small molecules and peptides into potential treatments for genetic diseases. And one of my focuses is on diabetes. So we're really excited about pushing that forwards. The research that we do is funded by many different funders over the years. I'm currently supported by the BBSRC and the Leverhulme Trust. And I'm grateful for all of the ability to do the work because of the funding that we've had. And the research group that work with me and my team are really important, and the drivers. And one of the best parts of my job is helping people in their career. So I'm very, very grateful for my research group and all of the work that we do together, in addition to our collaborators. So this leads me to the end of my talk, and I'm very happy to answer any questions that people might have.
Thank you so much, Zoe, for that really fascinating insight into what DNA is, how it exists, and how the widely recognized DNA double helix isn't the be all and end all, and how these peculiar knotted structures are really changing the way that DNA behaves and controls our biology. So thank you so much. It was brilliant. So we have, as Zoe said, a few moments now for I think about 20 minutes maybe to look at any questions that have come through on the Slido. So I once again encourage people who are online live to submit those questions, and we have a few, so I'll make a start if you're ready, Zoe.
Yeah.
So you've mentioned this a little bit already, but could you give us a little bit more detail on how these quadruplex structures, eye motif and G quadruplex, relate to diseases? Are there specific examples of diseases that are known to be in which these structures are implicated?
Yes, so there are a lot of work in the literature so far has focused on looking at the relationship of these structures in cancer. And I quoted 43% of genes contain a quadruplex forming sequence in. But shortly after that study was published, there was another one that indicated that 69% of oncogenes, so cancer-causing genes, contain one of these structures. So there's been a lot of focus on that because it looks like there might be some relationship between the two. I'm interested in diabetes because there's a lot of different sequences in the insulin gene. So that's one of my areas of focus. But these are prevalent in also areas for neurodegenerative disease, and quite a lot of others that are collected. But I think most of the literature is focused on cancer. I think in terms of trying to improve chemotherapeutics, because I think when people think of chemotherapy, they think of people losing their hair, and if they can make something a bit more specific, perhaps that will then reduce the number of side effects. So I think that's been the main focus, but there's many different conditions that could be affected by these structures.
Very interesting. You mentioned one type of bacterium there that's involved potentially in climate change. How widely do these structures spread across the different organisms that we find on planet Earth?
So they are highly prevalent, and I think that what's interesting is that bacteria have an evolutionary advantage, that if there are no barriers to their replication, because they replicate fast, and the fact that these structures take longer to unfold mean that if the bacteria are keeping them there, they must have some function. And so the presence of these sequences that can form these structures are observed in so many different organisms, and some of them you can map through the genetic lineage, and they haven't changed either. So they're obviously kept and retained. So I mean, the current hypothesis is that they might be the place where proteins will interact, and that then changes the way that we think about how genes are read. But yes, they're in lots and lots of different organisms. Some have more than others, and but they, largely are very prevalent.
And what about RNA, the photocopied recipe book? Does that
Yes.
also display similar topologies, structures?
Yes, and RNA does form it's known to form lots of different structures, and RNA is a very interesting molecule because it can do so many different things. And yes, these structures do form also in RNA, and they will have a different functional role in that because of it being a working molecule that gets broken down quite quickly. But yes, they're definitely prevalent in RNA, and it looks like that it might be able to affect the way that RNA is then used to then make the protein.
Yeah, very cool. I have a question here about when would we be able to turn off DNA relating to diseases such as Huntington's, and do these structures have any role in that regard?
So in terms of turning off genes, there is, I think, with what we're looking at for these structures is that they are a node where a protein will interact. So rather than switching it off completely, you're kind of turning it down more than completely off, I would say. It's more like a dial, perhaps. Perhaps I should change the way I talk about that. But I think with particular conditions such as Huntington's, where there is a mutation there, then these particular structures will affect them, but that wouldn't be the way to then solve that particular problem there. So we would be looking at if you needed a gene switched on more, then that's the way that could happen. But if you wanted to eliminate something completely, it would be better to use a different method, and there are plenty.
Got a really interesting question here about the metal ions that you talked about. So it says, "Hi, so great talk. Why only copper and silver would form the eye motifs? Do you know anything about the mechanism?"
It's a nice question. It isn't just only those metals, and we haven't done, and perhaps we should do a thorough going through every single possible metal. We did do a bit of a screen at the time, and we have looked at others. So I have a student that's just finished looking at iron. So we do have some work there. And then for the G quadruplex, it's known that potassium cations and sodium cations help stabilize those structures. So it isn't just those metals, those are just the ones that I wanted to highlight, but there's still potentially lots more that could be done with that. And in terms of the mechanism, the hypothesis with the silver cations is that it helps mediate the cytosines to come together. So for a cytosine to base pair with another cytosine, typically we need a little bit of acid. So this is the reason why they're pH responsive. And the hypothesis is that the silver helps those to come together instead of acid. So you can do that at neutral pH instead.
A DNA glue of sorts.
Yeah, of sorts.
Yeah.
And I have people very interested in that type of thing, and it is fascinating. But there's still so much potential with the metal cations.
I wonder, there's another one from Jane that says, do you think that A and T will have a similar activity? I think in regards to forming these structures, or are they very simple base pairs that don't engage in this activity, this folding?
Yes, so there are a lot of different structures that DNA and RNA can form, and I've focused on ones in GC rich in my career, and there are others. So, and if you have sequences rich in adenine, they can form a structure called the A motif, for example, but this will only form when you've got adenine under acidic conditions and it protonates both adenines. Well, apparently, I fully think that actually it probably will form in some way. Doesn't necessarily need to be completely acidic, but there are other structures that these will form. But the ones that I've focused on in my career have been in the GC rich regions.
Great. Another question here. Are there known protein partners for eye motif binding? And the follow-up is, has just moved. Do you know how any modifications of the DNA, I think, affect the structures that they form?
Okay, yes. So in terms of, are there proteins that bind eye motif? Yes, there are. There are a number of proteins that have been shown to interact with eye motif, and one of the researchers in my group is looking at one that we think stabilizes eye motif at the moment. So that's quite interesting and something that we're looking at in more detail. And in terms of modification, so is that modification of nucleic acids or modification of DNA?
I think it's modifications of the DNA itself, because I think there are certain changes, temporary or maybe even inherited, to the DNA bases themselves that maybe affect the folding.
Yes. So there is, in addition to our four bases of DNA, our genetic code, there is a whole other layer called epigenetics. So epi means above, like the sequence, so epigenetics. And one of those is in terms of methylation of cytosine. Methylation of cytosine is like it's like a bookmark. So it marks in the DNA. And we've shown that the methyl cytosine stabilizes eye motif. It can also, and what we're seeing is that the presence of a methylated cytosine can affect the dynamics of the structure. And what that means is that the proportion at which it will stay in an eye motif rather than another structure. But that's something that we're looking at. It is really interesting, and yeah, I've just got a little bit excited about that. But yeah, so it's something that we're looking at.
think you covered this a little bit, but maybe a little bit more detail to answer the question, which diseases is your team most interested in?
So I think the field is quite interested in cancer, as I've said, and I've been very privileged to work on the insulin gene for a long time. I've been funded by Diabetes UK and the Wellcome Trust, and also using the using the insulin gene as a model, also the BBSRC. So for me, I think diabetes is the main target for my group at the moment, alongside cancer. But I am really interested in looking at neurodegenerative disease, because especially at UCL being such a hub for neuroscience, it's really important to make the best of the environment you're in. So I'm really looking forward to looking more into that as well.
Very good. So we've got an interesting question where I may have to display a lack of knowledge of a certain scientific term. I'll read it as we go on. So if silver cations alter a gene in a plant, so you already admitted that the plants often have very, very large genomes, is it likely that this is due to eye motifs? And I think this is an example. Silver cations induce monoecy, never studied botany, unfortunately, in certain plants on a specific synthetic pathway. So I think the question is, do you think that this known change of a plant's behavior after treatment with silver could be due to eye motifs?
Could be. That sounds really cool.
I think that you've got a new collaborator potentially.
Yeah. Send me an email. Send me an email.
Whoever that is, where
Yeah.
Zoe works, so it would be great. I think Zoe would welcome that email. And yeah, there are some others that are largely covering what's okay. It's I think somebody's defined this term. It's also known as hermaphroditism or hermaphrodite. So I think that's where you have both male and female parts of a plant.
That's very interesting. Yeah, definitely worth looking at that.
To pick up on the protein binding partners that that you mentioned earlier, is if you were to target that as a therapeutic strategy, would it be you'd be presumably looking to change the way that the eye motif binds with that protein, switching it on or off depending on the biology? Is that right?
Yes. So I think the way that I see it at the moment, my current hypothesis is that if you're changing the shape of DNA, then the recognition point of the protein is then altered, because then it won't be able to interact in the same way.
Okay, so you could that's dial, or the switch, the dial that you mentioned earlier. You could, like the thermostat in your house, you could turn it up or down by potentially changing its interactions. Okay, that's very interesting. And in terms of the translational work that you've mentioned there with the peptides and the small molecules, where are you in that process? Have you started to test these in animal systems yet, or is that something that's still to come?
Still to come, yeah. So I think it's important to always do as much work in vitro as possible, and we have quite a highly specialized models, in particular for the insulin work, using special beta cells that we can use. But yes, that would be the next stage for what we need to do to make sure that things are safe and effective for then potentially for human trials.
And just one more from me. You showed the structure that your colleagues published in 2002, I think, on the G quadruplex. And I think you've published some structures on the eye motif as well, to look at exactly how they form together. How widespread are these structures of the eye motifs, and how different do you think they may be, to draw parallels with the way that we can target individual proteins in a cell, but that's not really done with targeting individual DNA structures? Is there lots of work still to be done there?
Yes, there is actually. There are quite a lot of crystal structures and a lot of structural information on G quadruplexes. And we've had plenty of time now since the first crystal structure in 2002. There are lots, and both Gary and Stephen and Joseph have done a lot of work in that area. Actually, we've been quite privileged to be involved in that at university level. In terms of eye motif, there's a lot less, and so the structure that I showed on that slide of the eye motif, that's the first intramolecular, so formed from one sequence in the field, and really we need more we need to do more, and there's not that many people working on eye motifs, but obviously I'm a big eye motif champion, but we're on it, we're working on it. And I think then once we have more data, so more examples of different ones, then we can then better predict. So then we can use prediction models, because at the moment then the models for perhaps proteins are quite well established because there's lots of proteins. And the models for DNA aren't as accurate, but with more actually physically crystallizing more structures, those will be able to become better. But we need that more data. But it's not easy to get those.
And then we can put the AI to work.
Exactly.
Presumably to help to predict things better. We've got one final question and a little bit of time remaining to maybe answer it. So do you see the treatment for diseases of this kind to be a medication that would be taken in the long term, or maybe a one-off treatment of the patient's DNA?
That's a really good question, because I think if you'd asked me maybe a year ago, I would have said, oh, that a treatment. But actually, I'm thinking probably both moving forward. I think we can probably, if there's the potential to be able to do a single treatment to change that DNA sequence. But we need to do more work on it. So I guess that's my job to do that. But yes, both probably.
Fantastic. Thank you very much. So we're coming up to the 1 hour mark. So I'd just like to say I thought your talk was absolutely fascinating. Thank you so much for kindly agreeing to speak about it. Before we close, if you'd like to explore more of what's happening during UCL's bicentenary year, there are several ways to get involved. To all those who are watching live on YouTube or watching later on, you can book a place on the UCL 200 walking tour. You can discover our full program through the UCL 200 events calendar and visit the UCL 200 exhibition on campus, which is running throughout the year and is a real must-see. It's quite something to visit and experience. We also have 3 new UCL 200 publications, and all of these are linked on the UCL 200 web pages. I think, Zoe, if there's a UCL QR code that maybe we could share, or the tech team could help us to share, so those online could to click onto that to find out more about the history of UCL, what we're doing now, and what we hope our university will be able to grow into and deliver in the future. But I think we've reached our time limit now, so thank you once again, Zoe. It was really fascinating to hear from you. Thank you everyone online for joining, and we'll see you again at the next event. Thank you. Bye-bye.